With this video, I'd like to continue explanation of causes of the lactate threshold and explain the lactate shuttle, and also introduce a technique called tracer methodology. And again, some of these notes I've copied from the American College of Sports Medicine Advanced Exercise Physiology textbook. So what is the metabolic fate of lactate? Well, one fate of lactate is based on the lactate shuttle hypothesis developed by Brooks in 1984. And this shuttle hypothesis is that lactate can be moved or shuttled around the body from the site of production, for example, a fast twitch muscle fiber, and is moved around the body where lactate can be oxidized, can be used as a fuel source or substrate in another cell. For example, a fast twitch muscle fiber. And this very simple diagram is shown on the next slide. And here we can start with a fast glycolytic or fast twitch fiber, where glycogen is broken down to glucose 6-phosphate and broken down to lactate that can then leave this muscle cell, get into the circulation. Lactate is oxidized by the heart muscle. The heart prefers to use lactate as a fuel. Lactate can also go to another muscle fiber, a separate slow twitch muscle fiber, where pyruvate then, lactate is changed back into pyruvate by the LDH heart form of the enzyme. And then that pyruvate is sent into the mitochondria, and that lactate is, or pyruvate from lactate is oxidized. Some updates to this shuttle hypothesis is notice that this lactate can go into the circulation, but also this lactate does not have to go into the circulation. It can go to a neighboring muscle fiber and not even get out into the circulation. So this is a diagram describing the lactate shuttle, and one new update where there's cell-to-cell lactate movement. Part of this lactate shuttle is also lactate going to the liver cell, and this is called the Cori cycle. And here lactate is not really metabolized or oxidized in the liver. Instead, in the liver, it's combined to make new glucose or glycogen by the process of glycogenolysis. So this is the Cori cycle going where lactate leaves a muscle cell, goes into the circulation, and goes to the liver to make new glucose. And Brooks was one of the first to show that lactate can be consumed, can be used as a fuel source in the leg muscle cell. So the leg lactate release declines with exercise duration, and the working leg becomes a lactate consumer. It actually will oxidize and use that lactate as a fuel source. And so what is this metabolic fate of lactate? Well, most of it during exercise is oxidized. It's burned as a fuel. So maybe 75% is oxidized by the heart. The liver will burn some of it, and by slow twitch muscle fibers. During recovery, it continues the oxidation in those fibers. It's converted to glycogen. The carbons are incorporated into amino acids. And the lactate metabolism depends on the metabolic state. Is it during exercise or during recovery? And this is shown nicely in this next graph that was from some of the original data of the lactate shuttle. And here is the percent of radioactive lactate that was injected into, these were done in rats. And the condition, and this is percent, so it's from 0 to 100%. And here's rest, continuous exercise, and intermittent exercise. And since this is a radioactive labeled carbon on the 3-carbon lactate molecule, the investigators could capture that radioactive carbon and measure the radioactivity. So at rest, most of the carbon was in carbon dioxide, so it was expired CO2. That means that it had to be oxidized. So lactate went to some tissue where it was oxidized, some into bicarbonate, some into protein. Here with continuous exercise, maybe 50% is oxidized, maybe a little bit less is oxidized during intermittent exercise. Some get stored as glycogen, some is as blood glucose, some remained as lactate. So this is the fate of lactate after injection of radioactive lactate. And so this is an example of using tracer methodology. So tracer methodology will use different isotopes of carbon. And that example was radioactive carbon. There's also C13 carbon and hydrogens, but hydrogen, like 2 hydrogen. There's a pulse injection technique where the labeled lactate is added to the blood in one single bolus, so one single concentration. And it's looked at over time, and the rate of concentration decline represents the turnover of this lactate. A continuing infusion, the labeled lactate is added at an increasing rate and until an equilibrium point is reached and the lactate appearance is equal to the lactate removal. And so I've copied 2 pictures from a textbook, and I'll try to briefly describe these pictures. You can read the figure legends as well. And they try to describe both the pulse injection and the continuous infusion. So this picture shows the pulse injection. So here, the authors of this book describe a barrel. And here's the barrel. Here it is on the left-hand side. Here it is on the right-hand side. And so here's this barrel. And this barrel, then there's a pulse injection of some tracer at time point 0. And as there's metabolic production, as there's entry into this barrel of water, they say down here, and then we measure the metabolic removal. We measure how much tracer is at the exit. Let's say, for example, over here in X, at point X, it's going to be diluted. So this is called pulse injection, and here's the log color intensity or the amount of the dye over the turnover. This next figure shows continuous injection. And here's continuous injection or continuous infusion. So now it's continually, or starting here, it's continually infused, and it's continually infused, and now this principle, the color intensity at time at this time point 0 increases until it reaches an equilibrium. For example, in this barrel letter C, and here's an example of measuring, the rate of the slow or rapid or no rate of turnover. So this is continuous infusion. The previous graph was primed infusion. And so the turnover rate is equal to the appearance minus disappearance. So that's the turnover of this lactate. And the RA, the rate of appearance of lactate, will depend on the volume and the arterial blood lactate concentration. The rate of disappearance, so the rate of appearance minus that arterial lactate concentration. And the metabolic clearance rate is the RD, the disappearance, divided by the lactate concentration. So calculates a lactate clearance rate relative to an arterial concentration. So this metabolic clearance shows that the rate of disappearance is dependent on this lactate concentration. And that's shown here as well. So the metabolic clearance rate, the rate of disappearance is slowed as blood lactate concentration increases. And the rate of disappearance is dependent on blood lactate concentration, and the rate of disappearance is a function of blood lactate concentration and the rate of disappearance. So this is the classic graphs we've seen before. And so now, if we look we look at this one on the left that's looking at percent of VO2 max and blood lactate, there's a difference between the untrained individual and the trained individual. And so the trained individual has a lower lactate concentration, and since metabolic at a given exercise intensity, and since metabolic clearance rate is dependent on that concentration, the clearance rate is going to be greater as the concentration is less. So one physiological adaptation with training is that they are better at clearing that blood lactate. And as we described in earlier videos, the trained individual has a lower rate of appearance of blood lactate. So at a given exercise intensity, their blood lactate concentrations will be less. So this has been a video of some causes of the lactate threshold, describing the lactate shuttle, and then we ended with just a brief introduction about tracer methodology.